Hello, my dear students. How are you all? I hope all of you are doing well in your life and in your studies. So we are just going to do the quick revision of the biotechnology principle and processes. So I'm Rashmi Mamgai. Welcome you here at the Physics Wallah Gulf channel. And today we are just going to start the quick recap of this chapter. But before I start the session, let me tell you a beautiful quotation. Knowledge is a treasure, but practice is the key to it. So as the quotation says that obviously it's important to gain the knowledge, but most important is to practice it in order to become so perfect for your NEET exam. So you have to practice all the chapter very nicely, and you have to do a lot of question practice, a lot of question in order to become perfect to get the seat in medical college, right? So let's proceed with the chapter biotechnology principles and processes. So according to European Federation of Biotechnology, they have defined biotechnology beautifully. They are like that in biotechnology, we are using the product of microorganism, or we are using them for our benefit, right? So according to, you have to remember this definition. According to European Federation of Biotechnology, they have defined biotechnology that it is integrated science. Integrated means we have integrate a lot of thing here. Integrated use of natural science means natural capacity of the microorganism to form the product. Natural sciences, and we are using living organism also. We are using their genes also in order to obtain the product, right? And living organism cell, or we are using their enzymes, or we are using their molecules, right? Or we are molecules means their genetic material, right? Or for the product and services, for produce products and services, and we will sell it in the market. So I'm sure you have done the chapter which is known as microbes in human welfare. So in the microbes in human welfare, we are using microorganism to get the product and selling it in the market. Like we're using them in the bakery, we are using them in beverage company, right, industry. So we are using them for a various purposes. And here in this chapter, so that is the part of traditional biotechnology. Traditional biotechnology means we are using the microorganism in order to obtain the product. But in the modern biotechnology, we are just playing with their genes. So we are cutting their gene, the gene that is giving the product that we want, right? So we will cut that gene, we will put it in the desirable host, and that host will make a lot of the copies of that gene, and that gene will undergo transcription, translation to form the product. And after forming a lot of the product, we will sell it in the market. So biotechnology, according to EFB, biotechnology are of two types. One, we are using their natural capacity, and other one, we are using their molecules, like their genetic material, right? So we will see that biotechnology are of two types. They are mentioning biotechnology of two types. Modern biotechnology. So we are just seeing the types of biotechnology or biotech, right? So modern biotechnology, and other one is the traditional or conventional biotechnology, as the name suggests. In conventional or traditional that you have seen in the microbes chapter, that is the traditional biotechnology, where we are using the natural capacity of microorganism to obtain the product, and we are selling it in the market, right? So that means it's the based upon natural capacity, based upon natural capacity of organism, of microorganism to produce certain products that we have seen in the microbes chapter. But in the modern biotechnology, we are using their gene, and we'll see what we will do with that gene, right? So we have a lot of instruments here. So it is involved, this technique is involved in use of genetic engineering. Genetic engineering, right? So we are using the genetic engineering. And where do we use this feed? So we use in the in vitro fertilization. So in vitro fertilization, test tube baby in reproductive health, that process also require the knowledge and technique of biotechnology or genetic engineering, right? Next is the synthesis of new gene and using it for our benefit. Next is the, treating certain diseases. So suppose a person having the genetic disorder, we can treat it also, right? So it is also used to treat the genetic disorder, or we can just, correct the disorder. So we can correct the mutation at the genetic level, at the embryonic level, right? So when we get to know that embryo has some genetic disorder, we can replace the defective gene with the good gene, with the normal gene. So yes, that is also possible. Correcting the defective gene, treat the genetic disorder at the embryonic level is actually possible, right? And synthesis of DNA vaccine. So one of the best example of DNA vaccine is corona vaccines, right? So synthesis of the DNA vaccine is also the field of biotechnology. Synthesis of DNA vaccine. So this is about the modern biotechnology, and we will see it in the application part. How do we use it? How do we apply the biotechnology? But here we will understand the principle and processes involved in the genetic engineering, right? So we are going to see the modern biotechnology here. So when we talk about the principles, what are the principles that we are using in the biotechnology? So first principle is genetic engineering. We are using the DNA, genetic material, and that suppose the DNA have a specific gene. So the functional part of DNA is known as gene, and we are using this gene will undergo transcription, translation to form the protein or enzyme. So suppose that protein or enzyme we need to sell in the market as a for the medicinal purposes. So we will just take out this gene from the DNA, and then we will let this gene to form the product in the host. So we use the host organism also. So inside the host, it forms the product, right? So product can be protein or enzyme that we want from out of this gene. So we will see the application later, but this is the genetic engineering, means we are just altering the DNA, right? We are just, taking the gene and putting it in the host, and inside the host, it will form a lot of copy, means or give the product. So cut, copy, paste. We will cut the gene, we will paste it in the host, and then it will replicate and form the copy of the gene and will form the product also after translation and transcription. So that you must have done in molecular base of inheritance, right? So we will see that genetic engineering is nothing but the cut, copy, paste. How? I will tell you, right? So the gene that we want, we will cut it, we'll copy it, and we will paste it. How? We will see. This is the first principle. Second principle is bioprocess engineering. So we are working at very micro level, at the microorganism level, because host is also microorganism, right, in which we will put our desirable gene to form the product. So that is also micro level. So we don't want any other unwanted microorganism to grow in the culture. So suppose this is the culture and these are the host carrying our gene, and suppose some contamination happen. So contamination means other microorganism has entered the system that we don't want to grow. So our product will be disrupted. So means we want sterile condition. Sterile means we don't want any contamination, non-contaminated system. So that is known as chemical engineering, and then we will produce the product, right? So we will manufacture the product. So we will ultimately form the product. That is our purpose. So let's understand these two principles you have to remember for exam. Let's understand what is genetic engineering, what is cut, copy, paste. So I'm just telling you the gist of the whole chapter, and then I'll start with the processes, right? So in the processes part, I'm just going to talk about the cut, copy, paste in the processes part, right? So when we talk about the processes of genetic engineering, listen to me carefully. When we're talking about the processes of genetic engineering, right? So in that, we will suppose this is the cell of the organism. Suppose we like the gene which is giving the high productivity in a plant. So we want that gene from that plant. Suppose that plant, that gene is responsible to form the particular type of protein, and which is very, very useful for us. So we just want that protein in huge amount. So what we will do, we will take the plant cell, right? Then we will just, destroy the plant, all the part, and just filter the DNA from that, right? Separate the DNA. So suppose this is the DNA of the plant. And then this DNA has our gene that we want. So in this DNA, suppose this is our gene. Gene is the functional part of DNA that we want. So suppose this is our desirable gene. Desirable means suppose this is what we want, right? This is our desirable gene. So we have to cut it from the DNA. So how do we do that? By using scissors. So we are having special type of molecular scissor or restriction enzyme. So we will cut the desirable gene, right? So we will cut the desirable gene. So we will cut the desirable gene, right? And after cutting it, cut the desirable gene with the help of molecular scissor. And what is molecular scissor? We will see it in detail. Restriction enzyme, right? Restriction enzyme. You have to remember this. Now suppose this is the carrier. So we require carrier also, gene carrier, right? Which is also known as vector. So vector, like mosquito is the vector for the malaria pathogen. Same way, vector means is the carrier of our gene of interest. So vector can be plasmid, and I'm sure that you know the plasmid, right? So we will cut it with the restriction enzyme. We will cut the vector with the restriction enzyme, RA for restriction enzyme, right? With the same molecular scissor. We will use the same restriction enzyme as we have used here, right? So we will cut it with the same restriction enzyme as we have used here. And now after cutting it, we will paste our gene of interest here. So we will paste our gene of interest here with help of glue, molecular glue. So we have pasted our gene of interest here. Gene of interest, right, is pasted here. So now we have got the recombinant DNA. What do you understand by recombinant DNA? Recombinant means it has the new combination. It has its own DNA sequence, plus it is having the gene from the outside. So that's why we called it recombination. A new combination has formed. So we have pasted it with help of ligase enzyme. So ligase enzyme will paste our gene of interest. After forming the recombinant DNA, what we will do? We will put this recombinant DNA inside the host, right? So we will put it inside the host by various methods that we will see. So this is suppose our host. It is having its own main DNA. And now we are putting our recombinant DNA inside it, so that our gene of interest will replicate, will form the copy inside the host cell. And this process we call transformation, because we have transformed the host. Transformed means we have changed the host by putting our recombinant DNA inside it. Now this host will replicate, and at the same time our gene of interest will also replicate and form the product. After replication, transcription, translation will happen inside the host of this gene, and it will form the product, right? So finally we will form the clone, right? So we'll form the clone means copy. Cloning of, we will form the clone of gene of interest. And then this will undergo replication, transcription and translation to form product, to form huge product, so that we can sell it in the market, right? So this is our purpose, and these are the processes. These are the step of the processes. We have cut our gene of interest, paste it with the cloning vector, and then we put that vector inside the host to form the copy. Cut, copy, paste. So these are the processes. This is the gist of the chapter. So let's see every step that I have told you in a very, very detailed way, right? So let me take you ahead. So when we talk about step of genetic engineering, I have told you we have to do the gene transfer. We have to do the creation of recombinant DNA, right? So then we will just make the copies of DNA, we will transfer gene into the host. This is what we have to do, as I have told you, right? So what are the steps? What are the processes of the genetic engineering? So first of all, what are the steps? First of all, we have to identify the DNA with desirable gene. And then what we have to do, let me just take a page. So first step will be identification of a gene. Identification and separation, right? Separation of DNA from the organism. Identification and separation of DNA. This is our first step. Second, these are the part of the processes of recombinant DNA technology or genetic engineering. So processes of genetic engineering, or we also called it recombinant DNA technology, RDT. Recombinant DNA technology, right? So after identification and separation, we will cut gene of interest. We will cut gene of interest from the DNA, right? From DNA. This is our second step. From DNA with help of restriction enzyme. By restriction enzyme. Third, after cutting them, we have to separate. We will get, after cutting this DNA, we will get a lot of fragments. So we have to separate our gene of interest. Separate gene of interest. So we have to separate our gene of interest out of all the fragments, right? So here we can use the appropriate term isolation. Isolation means to release out the DNA. After separating, we have to make the 1,000 copies of our gene of interest, right? So after that, we will do the amplification of DNA using PCR. After the PCR, we will put it in the host. That means transformation. And for the transformation, either we can directly put our gene in the host gene, we can incorporate our gene in the host. So we can do the direct transfer inside the host. Transformation means to put in the host. Either we will do the direct transfer or we will do indirect transfer or with vector. With help of vector, transfer with help of vector, right? Transfer of gene with help of vector. And after transformation, we will produce, we will grow the host in the bioreactors, which is the very, very large container. Why? To form the product. So 6th, 7th step will be, we have to, after forming the product, we have to purify it and we have to sell it in the market. That is the downstreaming process. So the whole chapter will be covered in the 7th step, right? Whole processes of genetic engineering and this part will covered in these steps. So these are the part of, till the 5th we called it upstreaming. Upstreaming means all these part. And then we will use the bioreactor to form the product. And after that, we will purify our product. So let's see these processes one by one in detail way, right? So the first step, what is the first step? That is the separation and isolation of the DNA using different type of cleaving enzyme, different type of lysing enzyme. So let's see what is it. So when we see the process of recombinant DNA technology, first process is identification and isolation of DNA. Identification and isolation of DNA. Isolation of DNA from the organism. So suppose we want the DNA, we want the gene which is present in the DNA of bacteria, or we want gene which is present in the plant cell. So we will use different enzyme to break down them and then isolate our DNA so that we can cut our gene from that DNA, right? So let's see what are the process. So first of all, we have to open the cell that the DNA of the cell that we want, right? So cell is broken up by lysing enzyme. So what are the different type of lysing enzymes for different type of organism? So suppose we are having the bacteria. So suppose bacteria, we are having the bacteria cell. So for that, which lysing enzyme will be used? Lysozyme will be used to break down the cell wall of bacteria, right? So lysozyme enzyme will break the cell wall of bacteria, right? And bacterial cell wall made up of the peptidoglycan. So obviously lysozyme enzyme will break down the lyticans, peptidoglycan wall. When we talk about fungi, so fungi cell wall is made up of the chitin. So this chitin cell wall will be broken down by which enzyme? So we are having the chitinase enzyme. Chitinase enzyme, right? So this chitinase enzyme will break down the cell. Break down chitin, which is forming the cell wall of the fungi. Same way, in case of the plant, we are using the cellulase to destroy the cellulose. So cellulose break down the cell wall of plant. So in that ways, if you are taking the gene from the plant, we will use this enzyme, right? And then we know that apart from the cell wall, we have the plasma membrane, cell organelle, different protein, RNA made up of protein, RNA, lipids and all. We don't want anything, right? So what we will do, we will just destroy all the macromolecule that we don't want and keep the DNA only there, right? So we will use various enzyme now. After this process, after breaking down the cell wall, what is the second step? Second step will be the proteases. We will use the enzyme proteases to break down the protein. We will use the enzyme carbohydrase, different type of carbohydrase to break down the carbohydrate in the cell. Apart from that, we will use the lipase to break down the lipid. We will use the RNase to break down the RNA. So that means we will just dissolve everything that we don't want, right? So all these things are used, all these enzymes are used to break down, to dissolve the other micromolecule, dissolve the macromolecule that we don't want. So what is left now when we have dissolved, when we have broken down everything, what is left? DNA only, because we have not used any enzyme to degrade the DNA. So finally, we will get the DNA, right? So how to separate the DNA? We will add the chilled ethanol in the system in which all this thing is going on. We are putting all this enzyme there in the system. And finally, in this system, after using all these enzymes, we will add the chilled ethanol, very chill, right? Chilled ethanol. So why we are using the chilled ethanol? So that it can precipitate our DNA. So our DNA will get precipitated. Why we have added the chilled ethanol? To precipitate DNA. So our DNA get precipitated, right? And now what will happen? Finally, using certain instrument, we will just take the DNA out. So it's like this. We will take the DNA out, DNA thread out. So this way, we will just put it out. And that is known as spooling. So spooling means collection of DNA, right? So after precipitating the DNA, we will collect the DNA. And process of collection of DNA known as spooling. So this is the NEET question. What is spooling? To collect the DNA from the organism, from the cell of the organism, from where we want our gene of interest. Isn't it interesting? This is the first process. So first process of biotech is done. Now the second step, using cutting up. Now we have collected the DNA, but we want our gene. So suppose this is the gene of interest. So we have to cut our gene of interest from the DNA. So second step will be using molecular scissor, using restriction enzyme, we will cut our gene of interest. So yes, we are having the restriction enzyme, right? So second step will be cleaving the DNA, right? Cleaving the DNA gene of interest by restriction enzyme. So we will see the cleaving by the restriction enzyme, right? Cleaving the DNA by restriction enzyme. So how does this restriction enzyme got discovered? We will see it first. You know, these two scientists, that is known as Arber and Wiener. So they were just, Arber and the, Werner were actually doing the work on the bacteria, right? So there are the two scientists, Arber and Werner. So these two scientists were actually working on the virus, right? Werner Arber and this, were working on working on bacteriophage, bacteria and bacteriophage is virus, right? So they were actually seeing that how bacteriophage virus infect the bacteria. They were seeing that they were working on bacteria infected by bacteriophage, right? So they were actually studying about them, that how bacteriophage virus infect the bacteria. And they found this so amazing enzyme in the bacteria as their defense mechanism. So what they have found, suppose this is the bacteria actually have a gene, suppose this is a special gene, and it is producing the special enzyme known as restriction enzyme. So what is the purpose of this restriction enzyme? Suppose this is the bacteriophage virus and it is putting its gene here to incorporate with the bacterial DNA, so that it can just after incorporating with the bacterial DNA, what it will do, it will replicate and form a lot of copies. So what we have seen, what they have observed that now this bacteria is producing restriction enzyme, and this restriction enzyme is cutting the DNA of viral DNA into various pieces. Viral DNA is destroyed. So they were so amazed that wow, bacteria has their own defense mechanism to destroy the viral DNA. That is really nice. And now some kids will ask me that, ma'am, this restriction enzyme can also destroy the bacterial DNA, no? But for their protection, they have other enzyme also known as methylase. And this methylase will actually, this is the other enzyme. So this methylase will actually do the methylation of DNA, add the methyl group in DNA, bacterial DNA, and just hide the area or hide the sequences that is recognized by restriction enzyme, so that they will not cut their own DNA. So they have the two beautiful enzymes. In this experiment, accidentally they have found this mechanism that this bacteria has two enzymes for their defense, right? What is that? First one is the restriction enzyme and other one is the methylase enzyme. So I have told you what is the purpose of methylase and restriction enzyme. So restriction enzyme cut the viral DNA, right? It will restrict the growth of virus in bacteria, that bacteriophage virus in the bacteria, by cleaving the viral DNA. So how it is preventing it? By cleaving viral DNA. So it is just cutting it, right, at the specific site. So this restriction enzyme recognize the specific site only, and that is also known as palindromic sequence or recognition sequence. There only they can cut the DNA. They just recognize some specific part. So they just found this in the bacteria. And methylase will actually do the methylation of bacterial DNA, so that bacterial DNA can protect itself, right? Bacterial DNA can protect itself. Are you getting it? So what it will do, it will hide the specific site. It will hide the specific site on bacterial DNA from restriction enzyme. So this is how they beautifully preventing them, but cutting the DNA of the virus only. And this is the defense mechanism, and it was the discovery of restriction enzyme was the turning point for biotechnology. So when these two scientists has observed it, they were like, oh, now we can just use the restriction enzyme as a tool to cut the DNA from the specific site, and we can directly paste it with the other enzyme or with the other organism where we want to grow or where we want to express the gene that we want. So in that ways, it was the revolution. Biotechnology has got the revolution in by discovering the restriction enzyme, right? So obviously, restriction enzyme is the nucleases. Nucleases means that will cut the nucleic acid, DNA or RNA. So nucleases generally two types, exonucleases and endonucleases. Exo means this is the DNA. So exo will cut from the end, So remove nucleotides at the end of DNA, and they don't require any specific sequence because they just act at the end, right? So generally this nucleases we don't use in the biotechnology. So we don't use it in the biotechnology. Do not use it in genetic engineering. We don't want it. And endonucleases means they will cut the DNA within. They will cut the DNA at the specific position in between or from within. And they require palindromic sequences. What is palindromic sequences? They recognize the specific sequence, which is also known as recognition sequence of the DNA, right? So it is also known as recognition sequence of DNA. Or sorry, restriction enzyme. So they will cut at the palindromic sequence. What is the palindromic sequence? Suppose, madam, so either you read this in this direction or this direction. So you will able to see that this is the same spelling. Either you read this way or this way, it is same. There is no change. So that is the palindromic sequence. So generally all the restriction enzyme have their own this kind of sequence, own palindromic sequence that will be same in both the direction. That we'll see ahead. And it help in the defense of bacteria that I have told you, right? It help in the defense of bacteria also that I have told you. So now I'm just going to talk about the endonucleases in a very different way. So in 1963, two enzymes were recognized in the E. coli. One added methyl group and other cuts the DNA, known as restriction enzyme, as I have told you. So, you know, HIN2 was the first restriction enzyme, very, very important. So HIN2 was the first restriction enzyme that was discovered, right? So how do we name them? Why do we calling them HIN2? So first letter of any restriction enzyme come from its genus, right? So first word actually represent the genus Haemophilus. So we have taken out it from the Haemophilus bacteria. IN represent their specific or species. That means influenza, IN from influenza bacteria, right? And this then we are having the D. This letter is actually representing the strain. So like we are having animal breeds, same bacteria has this strain. So D are coming from the strain. And when we talk about 2, so this 2 actually representing the order of discovery, right? Not discovery, order of isolation. So it's actually talking about the order of their isolation. Means where we have isolated this from the bacteria. Now you will say, ma'am, this is the first restriction enzyme that was restriction endonucleases that was isolated from the Haemophilus bacteria. So then why are we calling it second? It should be 1. It should be given 1 number. So you know what? The first enzyme that were taken out from the Haemophilus, actually it was not restriction endonucleases, it was some other enzyme, right? So means perhaps it was some other enzyme, like it can be exonucleases. So we actually talking about endonucleases. So this was the second enzyme but first restriction, second enzyme but first restriction enzyme. So it was the second enzyme because first enzyme was some other enzyme, not the restriction enzyme. But this is the first restriction endonucleases which were isolated from this bacteria, right? So that means how do we name them? First letter represent genus of bacteria from which it is isolated, and followed by species, its strain name, and 1 represent the order of isolation, right? So ECOR 1 is the other example. And nowadays, we are having more than, very, very important term for the NEET, I mean the statement for the NEET, more than 900 restriction enzymes are isolated from the 230 strain of bacteria. So means we have isolated more than 900 restriction enzyme from 230 strain bacteria, right? So when we have seen the nomenclature, we will see their recognition site now, how their recognition site look like. So some bacteria actually give us the blunt end and some give the, so how they actually work, right? So when we talk about their working, so some they produce blunt end, some produce sticky end. So example is written here that who are producing blunt end, And what is it? I'm just going to talk about it, right? Even Hindu is the example of it. This is also producing the blunt end. So let's discuss about it, right? What is the blunt end? So blunt end is the mode of action of restriction enzyme. Mode of action of restriction enzyme, restriction endonucleases. And we just use the type 2 restriction endonucleases. What is type 2? What is type 2? Actually, it recognizes the DNA and cut the DNA at the specific sequence, right? Or you can say it actually recognizes the specific site and cut the both strand of DNA. But when we talk about restriction enzyme 1, restriction enzyme 2, we don't use them, right? Restriction endonucleases 1, 2, 3, we don't use them. Only we use the type 2 NEET question. So restriction endonucleases type 2 are used, and these are the type 2 only. So as I have told you that they will just, recognize the specific sequence and mode of action, some producing blunt and some producing sticky. So what is the blunt end? It will cleave the DNA. They cleave DNA sequence from center. They will cut from the center, how we will see it. And when we talk about the sticky end, they will cut DNA away from the center. Away from the center of the recognition sequence of the palindromic sequences, because I have told you that they recognize the palindromic sequence, right? So you have to remember it. So when we talk about the blunt end, what is blunt end? Suppose this is the recognition sequence for enzyme, right? For the suppose SMA1. So this is the recognition sequence for SMA1. So if we read it from this direction to this direction or this direction to this direction, it is same. So GGCC, GGG, CCC. So that means this is the palindromic sequence. And this enzyme cut at the center of the recognition sequence and will give us this way, give us the DNA fragment this way, right? So means it will give the blunt end, means no single stranded, this is the blunt, this is the way we will get the restriction enzyme. So this is the blunt end cutting from the center, right? When we talk about sticky end, so the best example sticky end is 5 prime to 3 prime. We are having the recognition sequence GATTC and 3 prime to 5 prime NEET question. So CTTAAG. So either you read it from this direction to this direction or this direction to this direction. So they are same, right? So they are same in both the direction. So that means it is the palindromic sequence for enzyme ECOR1. So ECOR1 recognize this sequence and it will cut away from the center. So it actually cut between A and G. It will cut between A and G. So we will get this DNA sequence. We will get this DNA sequence this way, right? So we will get this product, we will get this one, and we will get this one. So that means we are getting the single stranded overhang. So you can see we are getting the single stranded overhang. So what is the purpose of these sticky end, which are the single stranded DNA or single stranded overhang? So these actually, because we are cutting, if we are cutting it with the ECOR1 enzyme, then vector that we are using to carry our gene in the host will also cut with the same ECOR1. So what we will get, we will actually get the same fragment, right? Or we will get the complementary fragment here so that we can paste our gene with the restriction enzyme, or sorry, with the vector. So if we are using ECOR1 here to cut this DNA and to this DNA, so we will get the complementary end. We will get the complementary overhang here so that we can paste it there with the ligase enzyme very nicely, right? So that's why these sticky ends are very useful for us, so that we can just have the complementary end with this. Complementary means AATT. AATT this side, TTA this side. So we can just paste our DNA fragment very nicely here with the vector DNA, right? So that it is going to help us to form the recombinant DNA. So finally, we have cut our DNA with the restriction enzyme. Third step or third process of the biotechnology is to separate the digested DNA. So suppose we have digested DNA, we have got many fragments, we have to separate our gene of interest. So for that, we use the agarose gel used to separate the DNA. And in that agarose gel, we have one end we are having the negative terminal, one end we are having the positive terminal, and we are just running the electricity from the terminals, from positive to negative terminal, right? So now these are the agarose gel plate. Agarose gel basically extracted from the weed. So agarose gel is extracted from weed, and it is mucopolysaccharide, right? Agarose gel is the polysaccharide. And we are, after this, after this agarose gel, after just warming up this agarose gel, it become liquefied, and then we put it in the electric feed or in this plate, agarose gel plate. So that gel, after cooling down, it settled down in a gel form, and we will make the well. By a little comb, we will make the well, and in this well we will put our DNA, right? So before putting our DNA in it, we will just add the ethidium bromide dye. So ethidium bromide is the stain. To use this dye is used to visualize the DNA, because DNA is transparent and also we cannot see with the naked eye. So after getting separated, how would we see them? So for this staining, we are using the ethidium bromide dye, which is carcinogen also, right? So we are putting the DNA now in the well, and we just submerge this agarose gel with the buffer system. Buffer is a liquid having different ions, and that will conduct electricity in one direction. So we know that DNA is negatively charged. DNA is negatively charged because of phosphate group, and they are put in the negative terminal, so they will just repelled by this negative terminal, right? And they will run very fast towards the positive terminal. But you know what? Agarose gel has a beautiful effect. What is that beautiful effect? Inside the agarose gel, we are having the sieving effect. Sieving effect means we are having the pores in the agarose gel, right? Sieving effect means we are having pores like chunni, like sieve that we use in our home to filter separate the flour, right? So that means sieving effect means it is having agarose gel having the pores in the agarose gel, through which our DNA, our charged particles will get separated. So any charged particle like DNA, RNA, protein can be separated on agarose gel. So you know what? DNA will run on through the pore, and the small DNA, smallest DNA will be able to run very fast through the pores. But the fatty DNA will not able to pass through the sieving effect or pass through the pores in the agarose gel. So that means the smaller DNA will run farther and faster, and large DNA will run very, very slow. So means DNA is actually separated on the basis of the size. So you have to remember that DNA in this process, that is in the gel electrophoresis, we are using agarose gel. In presence of electricity, it is running on the agarose gel, right? So DNA separated on the basis of size. On the basis of size means obviously smaller DNA, smaller pieces run farther and faster. Farther and faster, whereas the thick one will not be able to run so fast. So we already know the size of our DNA. Suppose this is the size of the DNA after getting separated under the UV rays, right? So visualize DNA under UV rays. So now after getting separated in gel electrophoresis, we will just put the UV rays here, and on the exposure of UV rays, it will give the orange band, right? So in the presence of UV rays, so we will use UV rays to give the orange. Because of ethidium bromide, right? It will give the orange band. Orange band of DNA is visualized. So why they become orange color? Because they are having the ethidium bromide, which is giving them the orange color in presence of UV radiation. And now finally, after we get to know that this is our DNA, we will just take it out from the agarose gel. So finally, we will separate our DNA from agarose gel. So finally, we will separation of gene of interest or desirable gene that we want. We know already the size of it, right? So this gene of interest is separated from agarose gel. So that means from agarose gel plate. So that process called elution. So if somebody asks you what is spooling, spooling is the isolation of DNA from organism or cell of the organism, and elution means after cutting our gene, we are separating it using electrophoresis. So after electrophoresis, we are just separating our gene of interest from the gel plate, right? Now, after we have separated our gene of interest, now we will form the multiple copies of it, right? We have to multiply it. We have to make 1,000 copies of it. So for that, we are using fourth step, that is the amplification of gene of interest. So amplification of gene of interest using PCR, and PCR for polymerase chain reaction, which was used by polymerase chain reaction, which was used by the Carey Mullis for the first time, invented by Carey Mullis, right? So you have to remember this. That is the polymerase chain reaction. So what is the polymerase chain reaction? Used to amplify our gene of interest in the 3 steps. So what are those 3 steps? And we are using PCR. We are using very, very high temperature in the PCR. Suppose these are our gene of interest, right? What we will do? So suppose this is our gene of interest that we want to amplify. The first step will be denaturation. So for denaturation means we have to convert them single-stranded. We have to separate both the strands. So denaturation means in the 94 degrees Celsius, which is again very high. So we have converted the DNA into single-stranded, right? So now they have separated and convert into the single-stranded DNA. Finally, we have done this. After this, what we will do, we will take the oligonucleotide or primers. In a natural replication, what you have used for replication? Primer, because we know our DNA polymerase enzyme cannot initiate replication. So for that, we require the oligonucleotide, small sequence of the primer. But here we use the single DNA, complementary DNA strands only, right? Single complementary DNA strand as a primer. You have to remember in this process, in the artificial PCR replication, we need the single-stranded complementary DNA, right? So obviously the second step, first step is denaturation at 94 degrees Celsius. Denaturation means DNA separated into single-stranded DNA, right? So now after this, we will do the second step, that is the annealing. We will do the second step, that is the annealing. So annealing actually requires 50 to 60 degrees Celsius. 50 to 60 degrees Celsius. So for that, we have to paste the primers. So we have pasted the primer, which is the single-stranded DNA, complementary DNA. We have pasted on both the sides, known as annealing. After pasting it with the help of the DNA polymerase, we will extend it. We will add the nucleotide here, right? So that is known as extension. So third step will be the extension step. So which is basically done in the 72 degrees Celsius, which is again very high. So normal DNA polymerase enzyme cannot be used here. So we use the special DNA polymerase enzyme known as Stack polymerase. Why do we call this DNA polymerase Stack polymerase? T for thermus, AQ for aquaticus. So we have taken this enzyme from Thermus aquaticus bacteria. So we have extracted it from Thermus aquaticus, is the special thermophile archaebacteria, right? aquaticus. So means it's a special it is thermophile. Thermophile means which is actually temperature loving. They live in volcanoes, so they can survive this will not get denatured at high temperature. It will be functional in the PCR machine, right? So that's why we are using the Taq polymerase with some other raw material like deoxynucleotide triphosphate, various type, right? We require deoxynucleotide, adenonucleotide triphosphate, adenosine triphosphate, and then we need the adenine, we need the guanine, we need the thymine, and so on, right? So we require all the type of deoxynucleotide triphosphate with adenine, with guanine, with cytosine, with thymine. And this enzyme also require magnesium ion as a cofactor. So after using all these things, we will add the nucleotide complementary here, and finally we will get our replicated copy. Finally replication is done. We have got the copies, right? So the same process will start again. So that means this step will start again. That is the denaturation, then annealing, then extension. This will keep on repeating. So we just get if we want to find out the copy of DNA, right? How to find out the copy, or suppose we have to find out how many copies of DNA we will get. Copy of DNA. Right, so formula is 2 raised to power n, where n is the number of the PCR. N is the number of PCR cycle, how many times we are doing the PCR. So suppose if we are doing PCR for 1 times, we will get the 2 replicated DNA, 2 pieces of DNA, as we have seen. Suppose we are doing it for 2 times, we'll get the 4 copies of DNA. And suppose we are repeating it 30 times, we will get 1 billion copies of DNA. 1 billion copies of DNA. So 1 billion times it get replicated. Sorry, 1 billion times, right? 1 billion times amplification we get. So you can imagine that we can get billions of the copies of our gene of interest. So this process, that is the denaturation. You have to remember the temperature used in denaturation, annealing, and extension. Very important for the NEET, right? So after this, finally, we will paste our DNA with the vector or put it in the host. So either we can directly put in the host or just do it with the help of vector. So let's see what we can do. So now finally, we can just transfer it in the vector or we can directly put it in the host. So let me take the other part. So we can either ligate. Ligate means paste. We can either do the ligation of gene of interest. Gene of interest with the restriction enzyme, right? Sorry, with this, ligase, gene of interest with vector, with cloning vector. Either we can paste it with the cloning vector if required. And then we will put it in the host. But before we put it in the host, we have to make it, we have to make the host competent, right? So finally, we have to introduce our gene of interest, introduction of gene of interest in the host. For that, we have to make the host competent, right? So means these are the step. After 4th step, we are having this 5th step, we are having the 6th step. So how to make the host competent? So suppose this is our host. Suppose this is the bacteria, which is our host. And we have to put our gene of interest with the vector inside it, right? We have to put our recombinant DNA in the host, suppose. So how to make it competent? So suppose it's a non-competent, it cannot take up any gene. It is not able to take up. Non-competent means not able to take up. So how to convert it into competent? So we will use the divalent ions. So what are the divalent ions? Like calcium ion. So we can use the calcium chloride solution, right? Or we can use the divalent ions. Divalent ion means like calcium ion, right? And we will give them heat shock. Means we will put them in high temperature and then we will do them cold, put them in the ice. So we will give the heat shock, cold or ice, and give the calcium. So this calcium ion, we know that lipid or carrying the negative charge, or this, membrane carrying negative charge, it will be coated by the positive charge. And so that our DNA, which is the negative, our DNA which is negative, will attract it towards the positive, and it will be able to take up our DNA. And by heat and cold shock, what will happen? Pores will created on the membrane, right? So this will create the transient pore. It will create the transient, means temporary pore, right? Transient pores on bacterial surface, on bacterial surface, so that it can take up our DNA, right? So that it can take up our DNA. Transient means for shorter time some pores are created to take up our DNA. So you have to remember, this is how we can make the host competent to take up our DNA. So finally, after making it competent by treating it with the divalent ions and heat and cold shock, we can put our DNA two ways, as I have told you. How can put our DNA two ways? So first way is direct transfer, other one with help of vector, right? So we can transfer the gene. We can transfer gene in competent host that we have made competent. So we have made it competent by treating it with this, right? So this is the competent host now. We have convert it into competent host. And in this host, we can put our gene of interest. So finally, there are two ways. One is the direct gene transfer. Direct gene transfer, we can directly transfer it in the host without using vector. And second is the vector mediated transfer. With help of vector mediated transfer. So let me tell you how can we directly inject our gene in the host. So first way is the using some chemical. So what kind of chemical we can use to directly take our DNA inside the host? So suppose this is host and this is our gene of interest. So we will just want to incorporate, this is our gene of interest. We want to incorporate it with the DNA of the host, and so that it will replicate and give us the product, right? So when we talk about chemical method, in this we are using treating it with the chemical. Treating it with chemicals like calcium phosphate, like calcium phosphate, or PEG, polyethylene glycol, so that we can make host more competent for, or make it high uptake, competent to uptake our, or endocytosis our DNA, so that it can take up our DNA very nicely, very easily. So we are making it highly competent for taking up or endocytosis of the DNA. This is the direct method. Second part of direct method is the second way is the micro injection. So as the name suggest, micro injection means, suppose this is the small injection, micro injection, and we are just putting the gene in the host cell with the help of this injection. And the best example of micro injection, it is used in the ICSI, intracytoplasmic sperm injection, in which single sperm is directly fused with the nucleus of the egg, right? That we have done in reproductive health. So best example is the intracytoplasmic sperm injection, means one sperm is directly injected or fused with the gene of the nucleus, or sperm is fused with the egg, right? So injection, it will be injection. Third way of direct gene transfer is the electroporation. Electroporation means we are giving the high voltage electric shock for very brief time to the host, so that it can take up our DNA and infuse our DNA with its DNA, right? So electroporation means we are giving high strength current, high strength current or electric shock, you can say, for a brief period of time. Brief period of time, approximately 1 to 2 microsecond, so that it can create the transient pore. It create pore on the surface of the host, and it will easily able to take up our DNA, right? Create transient pore in host so that it can take up our DNA, right? And third, next way is the gene gun method or biolistic method. Gene gun, as the name suggests, biolistic or gene gun method. So in this, we are just using the gene gun and just injecting our DNA coated with the gold and tungsten, which is very delicate element. And why we are coating our DNA with the gold and tungsten? Because DNA is so delicate fiber, and we are bombarding it with the high speed directly inside the plant cell, so that DNA thread can be broken down. So that's why, for the safer side, we just coiled it around the tungsten and the gold, right? And then bombard it in the plant cell mainly. So we just put it in the plant cell. This method is used to put the DNA in the plant cell. So microparticles of gold and tungsten are used to coat the DNA. Gold or tungsten coated with DNA. Coated with DNA is bombarded, are directly bombarded, onto the host cell, so that our host cell or host cell here is the plant cell. So you have to remember NEET question. They have asked this question that which technique is used to directly inject the DNA in the plant cell. It will be the gene gun or bombarded method, right? Or ballistic method. So that's all. This is the way of direct gene transfer. Now the other way, using the vector. So the third next way is the using the vector. So let's discuss about the vectors, different type of vectors that we can use. And what is the characteristic of ideal vector? NEET question, right? So how can we use vectors for the gene transfer? So the ideal characteristic of ideal vector. And what are the vector? Vector means a piece of DNA that is able to carry our gene to the host, right? So what are the vectors? Vectors is nothing but the piece of DNA. Piece of DNA that is able to carry, right? That is able to carry gene of interest in host. Whosoever is able to take up our gene of interest in host will be known as vector. So what are the characteristics? NEET question. What are the characteristics of vectors here? So you have to remember characteristics of ideal vector. First, it should be small size. It should be small size. Second, it should have origin of replication. So what is the purpose of origin of replication? NEET question. It is having two purposes. So if a host has origin of replication, it help it in the self-replication. Means it can replicate inside the host by itself. It doesn't need anything else for its replication. And second, it can also decide the copy number, right? It also decide copy number. Copy number means it has ability to replicate and how many times it is replicating. Suppose one vector is replicating 100 times and forming in 1 minute and forming 100 copies of the plasmid, and other have the capacity to form the 10 copies in 1 minute. So which is having the high copy number? Obviously first one have the high copy number. So we generally prefer the origin of replication of vector with high copy number, right? So they should have the high copy number, right? Third character is the restriction site. Restriction site means the site for the enzyme where we can cut it with the enzyme to paste our DNA. So we also called it cloning site. So restriction site, preferably, if this is the vector, it should have only one site for one restriction enzyme. Suppose this site for ECOR 1, then for ECOR 1, there should be only one site. If there will be many sites, what will happen? It can cut the plasmid and form many fragments, and it will become useless for us. So we just want one restriction site in this, one restriction site in the vector, right? So how many restriction site? One restriction site for one vector. So ideally, a vector should have one restriction site for one restriction enzyme. Then only we will call it the ideal vector. And the fourth character is multiple cloning site. Means, what does it mean? We will prefer the multiple cloning site. Multiple cloning site means they should have this character also. It will be good for us, right? So icing on the cake, it will be icing on the cake for us. So multiple cloning site means, suppose there is a vector. Suppose it is having the site for ECOR 1. ECOR 1 can cut it. Also, we can cut it with the PST1 restriction enzyme. We can cut it with the BAMH1 enzyme also. We can cut it with the SUV also, right? Or you can say PSV also. So in that ways, we are having the many site for different restriction enzymes. So we have greater choice now. We can use BAMH1. Suppose we want to paste our gene of interest at the site of PST1. We can use the PST1 site also. We will cut the restriction enzyme with PST1 and paste our gene here, right? So we have many options now. So many restriction enzyme, many different restriction enzyme, not the same, many different restriction enzyme can be used. So that means we have got wider options now. And the last is the selectable marker. Selectable marker means they are generally antibiotic resistant gene. Selectable marker are generally the gene that is making it antibiotic resistant, means they will just not allow the bacteria to die in the particular antibiotic because they have the resistant gene for them, right? So antibiotic resistant gene will also help us in selection of the transformation, the host that is having our gene of interest and the host that is not having our gene of interest. It will help in the selection of transformant from the non-transformant, right? So what is the purpose of it? Selectable marker will select transformant from the non-transformant. This is the purpose, right? Transformant host. Transformant means which is having our recombinant DNA from non-transformant host, right? So let's discuss about the different type of the vectors now. So we can use plasmid as a vector. So plasmid are basically present in the bacteria, right? So it's the extra, so the main DNA, the bacteria has main DNA plus extra chromosomal DNA, means they are having the extra DNA also, which is the circular self-replicating, double-stranded circular self-replicating extra chromosomal DNA in bacteria. Bacteriophage virus, also we just convert it for our purposes. Virus generally infect, cause the disease, right? By incorporating its DNA with the DNA of bacteria. They infect the bacteria, but we actually transform them. We just, cut their dangerous gene that is causing some disease and replacing it with our gene of interest, right? So that we are just cutting the viral DNA, the dangerous gene, and pasting our gene of interest there, making them so disinfectant, disarmed for our purpose, right? So bacteriophage also used as a vector. Next is the cosmid. So cosmid is actually the combination of two things. One plasmid, other one is the cos site of virus. So cos site, cohesive site of virus is pasted with the plasmid. We are actually having the combination of these two gene. Cos site gene of virus pasted with the plasmid, and artificially modified cosmid is basically produced here, right? And obviously which virus cos site from the lambda phage virus is used. So artificial vector like PBR322 and PUC1718 was designed, right? So that they can just serve the purposes that we want them to serve. So let's see all the type of vector in detail way. So this is the artificially produced vector, that is PBR322, P for plasmid, BR after the name of scientist, and this sequence represent the the serial number of the enzyme that is kept in the lab, right? So P for plasmid, B for Bolivar after the name of scientist Bolivar, Rodriguez, R for Rodriguez after the name of scientist, and this numerical value actually represent the serial number of this enzyme, or sorry, plasmid, serial number of plasmid in lab. So the lab in which we have kept it, we give the serial number to all the vectors. So this is representing the serial number, right? Now, this particular PBR322 artificially produced by these two scientists having all this thing, right? So it is having the site for PST1, PVU1. So see how many enzymes we can use, right? So BAMH1 and SAL1 restriction site for this enzyme present on the tetracycline resistant. Tetracycline resistant gene means this gene is responsible for producing the chemical or enzyme that will make it tetracycline antibiotic resistant. And I have told you that these resistant actually act as a selectable marker, right? So how we use it as a selectable marker, we will see it, right? These three restriction sites for restriction enzyme, ECOR1 can be used here, CLAV1 will cut here, HINT3 is cut here, right? So we can use any of the enzymes which is written here. Ampicillin resistant has two enzyme sites. PVU1 can be used here to cut the using the PVU1 restriction enzyme. We can cut this part and paste our gene of interest here. Even we can use the PST1 enzyme also. And this site, where these two site of restriction enzyme are there, is actually coding for the ampicillin resistant gene. So that means it will make the host resistant to the ampicillin antibiotic. No? And this ORI is origin of replication. ROP is the gene that is sequencing for the protein required for replication, right? So ROP is encoding for proteins required for replication. Required for what? Replication. Got it? So this is how the PBR322 look like. So let's see how do we use it. So suppose I have used BAMH1 enzyme to cut the vector PBR322. I'm using BAMH1, right, to cut this part. So suppose I have used BAMH1 enzyme and BAMH1 site is there. So cut the part of this this section. So tetracycline resistant gene is interrupted here. That means I have cut the BAMH, use the BAMH1 enzyme to cut the part. So I have cut the piece of the gene which were the part of tetracycline resistant gene. And now I will paste my gene of interest here. So suppose I have pasted my gene of interest here. So what I have done the insertional inactivation of tetracycline resistant, means now tetracycline resistant gene got inactivated. It will not produce any resistance to the tetracycline now, right? So suppose, let's see what will happen. So suppose desired DNA is cut from the BAMH1, and obviously plasmid PBR322, we also cut it with the BAMH1 because same restriction enzyme is used to cut the gene of interest and the BAMH and the plasmid vector, right? So now this desirable DNA is inserted, is basically cut, and even vector also cut. Vector and desired DNA cut with BAMH1. BAMH1. Now what we will do, because we are cutting it with the BAMH1, we will paste our gene of interest here at the site of BAMH1, right? So we have actually produced the recombinant DNA. So because of this, what will happen? Insertional inactivation. Insertional inactivation means because of insertion of our gene of interest, we have inactivated tetracycline resistant gene because our BAMH1 site was present here at the tetracycline resistant gene, and we have pasted our gene of interest here. So it has interrupted because we have inserted our gene of interest here. It has inactivated the BAMH1 site. Sorry, it has inactivated the tetracycline resistant gene site. So that means our recombinant DNA does not have tetracycline resistant gene, right? So what we will do, we will get the three things. We will do the transformation. Transformation means, you know, when we are doing this process of cutting it with the BAMH1, we will get the three condition. Some plasmid will be there which is not having any, which were protected by or not cut by the BAMH1. So they will remain as it is. As it is means no insertional inactivation happened in them. So we will get the two type of plasmid when we are doing this process. So some will remain as it is like this, and some plasmid who have got our gene of interest will get recombinant. So some will become non-recombinant, which doesn't have our gene of interest, and some will be recombinant, right? So when we talk about non-recombinant DNA means they don't have. So we can, because we are doing it at micro level, we are not seeing what is happening, we are putting enzyme only. So BAMH1 has cut some plasmid but has not cut some plasmid. So three condition after transformation means we will just put these plasmid inside the host. Transformation means transformation in E. coli. E. coli is the host here. So we will get the three condition. One E. coli without any plasmid. Suppose some host has not get any plasmid. So this is the non-transformant because there is no transformation. No transformation means no change, no plasmid has gone inside it. It is empty, it doesn't have any plasmid. Second case, second condition will be like, second condition will be like this recombinant DNA has entered in it. This has entered in it, so means it is having our recombinant DNA. And it is having plasmid, so we called it transformant with recombinant DNA. Transformant with recombinant DNA, right? And third condition, in the culture we have seen we are having the host but with the PBR322 without recombinant DNA, means it is having the non-recombinant DNA, means not having our gene of interest. So if it is not, if this PBR322 doesn't have our gene of interest, means no insertional inactivation in it. So in this particular PBR322, BAMH1 is also functional and ampicillin resistant, tetracycline resistant is also functional. Ampicillin resistant also functional. Both the genes are functional in it. But in the second condition in which our gene of interest is inserted at the BAMH1 site, tetracycline is basically got inactivated here. So that means it's also transformant. Transformant means this bacteria has the plasmid but not our recombinant DNA. So we don't want this and this. We want second condition. So how do we select second condition from these two? So for the non-transformant, we will add in this container, we will add ampicillin antibiotic. So we know that our required, this also has ampicillin resistant gene intact. It was not affected. And obviously this non-recombinant also has, it to have both the gene activated. Both tetracycline resistant, ampicillin resistant, both are activated in it. But it has the ampicillin resistant activated. So if we add ampicillin antibiotic, right? If we add ampicillin antibiotic, we know it doesn't have any plasmid. So it doesn't have any ampicillin resistant gene. It will die. It will not grow, right? So according to you, it will grow or not? It will not grow. Second condition has ampicillin resistant gene, so it will be able to resist the ampicillin antibiotic and will grow continuously. So ampicillin will not able to affect it, so it will grow, right? It will grow here. It will grow in presence of ampicillin. And third condition will also grow. So we have selected non-transformant. We have selected transformant from non-transformant in the first step using ampicillin antibiotic, right? So in the first step, we have selected the transformant from the non-transformant. So now we have to select the transformant with the recombinant DNA, means having our gene of interest. How do we do that? This is our recombinant DNA, and this is the another one which is not required. So we will put the tetracycline now. Tetracycline antibiotic. So this will not grow because we know that our recombinant DNA have inactivated tetracycline resistant gene. So it will not able to resist tetracycline, so it will not grow in this presence. But this is not having our gene of interest, so it is having ampicillin resistant also intact, and it is having tetracycline resistant also functional, so it will grow. So if it will grow, that means we know that we don't need this, we need this one in the second step. But generally PBr322 has drawbacks. We don't use this plasmid because it is very hectic process, very cumbersome process. That in first step you are choosing transformant from non-transformant. And finally in second step you are choosing a recombinant DNA with the non-recombinant DNA. Non-recombinant means which doesn't have our gene of interest, which we don't want. But it is hectic and it is like it requires 2 steps. So this is having some disadvantage. This is having some disadvantage. It is the cumbersome, means very laborsome process. Cumbersome process requires 2 steps. So we generally don't use it. Requires 2 steps to select the E. coli or host with recombinant DNA. Right? So we actually require this, and this is also very, very hectic process. So we generally don't use PBr322 plasmid. So second, the drawback of this is actually overcome by the second step. That is the second step, that is the, PUC 17 and 18. So I'm just talking about it. So when we talk about PUC 18 or 16, sorry, 16 or 17 or 18, let's discuss about it, right? So second plasmid is the PUC. Second plasmid is the PUC 17 or 18 we use. So P for plasmid, U for University of California. So it was developed in the University of California. So the name 17 or 18, this numerical value represent what? It actually represent the serial number of the of this plasmid in the lab, right? So PUC 18 actually like this. PUC 18, suppose this is the ampicillin resistant gene site, and this is the site. Multiple cloning site means having the restriction site for many enzymes, right? So we can use ECOR1, we can use BAMH1, we can use PST1, so it is present at this site, and this site actually code for LAC-Z gene. And LAC-Z gene actually translated into the beta-galactosidase enzyme. This gene is responsible for the beta-galactosidase enzyme. So what we do in this condition, suppose we have cut our gene of interest with the help of ECOR1. So we will just destruct the LAC-Z gene in recombinant DNA. Because our cloning site is present on the LAC-Z gene, and we have to cut, we have cloning site here only. So we will cut it with the enzyme, and we will paste our gene of interest here, so that LAC-Z gene will get inactivated, right? So let me take the example of PUC 18. Suppose we are having the desired DNA. We are having the desirable DNA. So we are putting this desirable DNA inserted into LAC-Z gene. So we are inserting it in LAC-Z gene site. And because we have inserted, because we are having cloning vector at LagZ gene site, so what will happen? Our LagZ gene get inactivated, right? So inactivation of LagZ gene will happen in our recombinant DNA. So suppose this is our P18 and we are having our gene of interest pasted at LagZ gene site. Now what will happen? We will transform the host. We will put it in the host. Transformation inside the host. Again, we will in E. coli, suppose we are using E. coli host, right? This is our host where we have to insert our gene of interest. So again, we will get three conditions. First, without any plasmid. Second, with the recombinant DNA that we want. And third with the normal, normal plasmid, normal packet without any insertional inactivation. Third one is the normal packet without any P18 without any insertional inactivation. It is recombinant DNA. It is non-recombinant, means it doesn't have our gene of interest, right? So means how do we select this non-transformant from the transformant one? So for this we will use the ampicillin, because this is the ampicillin is the selectable marker. Ampicillin resistant will remain intact in the condition, in the recombinant DNA condition, right? And it will obviously remain intact there. So it will not grow in ampicillin. It will grow in ampicillin. It will also grow in ampicillin. So both transformed will grow, non-transformed got eliminated by growing them in ampicillin, right? Now, how would we select the transformant with recombinant DNA and non-recombinant DNA? For that, we will use a chromogenic substrate. So now we will add chromogenic substrate XGAL, right? Substrate means some reactant which will convert into some different product and give the different color, right? XGAL. So we are having the two condition, one host with the recombinant DNA, other with the non-recombinant DNA, right? And non-recombinant means LagZ gene is intact, forming beta-galactosidase enzyme, right? So beta-galactosidase enzyme is produced in the non-recombinant DNA in the third condition. And this is the second condition, host. So now we will grow this host with the recombinant DNA in the substrate. We will put the substrate here. So it doesn't form no beta-galactosidase enzyme will be produced here. No galactosidase enzyme will be produced here. So substrate, chromogenic substrate, because we don't have any enzyme, it will not give any color. No color at all. But in the third condition, we are having the LagZ gene intact here. So that means it will actually convert the blue color. It will convert the substrate into blue color. So this enzyme will convert XGAL chromogenic substrate into blue color. So blue color means we don't want these colony, right? Color. That means we don't need the bacteria with blue color. We need bacteria with white color, because they are not giving any color, means it will seen as white, right? So if we see the Petri dish, if we see the dish where we are doing this experiment, we will get the two set of colonies. One colony with blue color bacteria and other colony with the white color bacteria, right? So we require the white colony. So white colonies has our recombinant DNA. Bacteria with recombinant DNA, we don't need the blue colony. So this is known as white and blue selection, right? White and blue colony selection. So we just select white one because they are having the bacteria with our recombinant DNA, right? Colony selection. So this vector is used here, right? That's all. This is done about the vector. Next type of vector that we use here is the the other vector that we use here for the plant, for the recombination of plant, is the TI plasmid, right? So suppose we are having third vector, that is the TI plasmid. So what is TI plasmid? Let me tell you. Suppose this is the plant, right? This is a plant having the leaf, right? Having the roots. And this is the dicot plant. So suppose their roots have some wound. There is the wound in the root and there is a soil-borne bacteria which is having TI plasmid, right? So this TI plasmid has the tDNA, tumor causing DNA, tDNA, which is causing the tumor causing DNA, or T for transfer, right? So this tumor causing DNA will transfer and inject, it basically incorporate with the DNA of plant cell. So if we see the root of plant cell, so suppose this is the DNA of the plant. So what will happen? Suppose this is the DNA of plant, right? Suppose this is the DNA of plant. This way. So this tDNA get incorporated with the plant DNA, right? And just this tDNA will produce huge amount of cytokinin or auxin. So that continuous division will happen. We know this two hormone will do the continuous replication of the bacteria, right, of the cells. So suppose when there is a wound, this tumefacient bacteria which actually infect the plant, right, plant cell. So tumefacient bacteria will enter the plant with the wound, through the wound, right? So obviously this is the soil-borne bacteria which present in soil, and it just infect the soil by just entering the TI plasmid in the cell, and just this TI plasmid paste its tDNA with the DNA of the plant cell. And we know this tDNA will produce a lot of cytokinin auxin, and plant root cell of the plant keep on dividing to form the tumor, right? So this is called as the crown gall or tumor, which will cause in the dicot plant mainly. So this bacteria normally cause this disease. Scientist has observed it. So what scientist has done, they have replaced this tDNA, tumor causing gene with the gene of interest. And then they have pasted it here at the site of tDNA and just letting the plant cell infect with the TI plasmid. And now this tumefacient bacteria is disarmed, means their tumor causing DNA is converted into is basically replaced with the good our gene of interest, and now it is carrying our gene of interest inside the plant, and plant will give us that expression, produce that character that we have put the gene on, right? So now you just see that what will happen, we have disarmed this bacteria. We have disarmed means we have replaced by replacing tDNA with gene of interest. Suppose this gene is for good productivity, good food production, right? So gene of interest. So we have replaced our tDNA with gene of interest, and now this gene of interest will be incorporated with the plant cell. So using the agrobacterium tumefaciens, this gene of interest will be incorporated with the plant cell, and when the plant cell will grow, it will give us the benefit that we want. So means we have converted this pathogen into disarmed, means we have taken the dangerous gene from the bacteria, from the TI plasmid, and then we just replace it with the gene of interest, and then we let the bacteria infect the cell and put the gene of interest, transfer the gene of interest with the plant cell. So now this plant cell has our favorable gene, right? Same way, the retroviruses are also used as vector. So these are also, we just do them disarmed. We just cut the gene that is responsible for the cancer and we replace it with the gene of interest. So these are also disarmed vector. We just disarm them, convert them for our animal cell, right? So we can put the gene in the animal cell, the gene that we want, using the retrovirus as a vector. So that's all about the insertion of our gene inside the vector. So what we have done, we have, suppose we require gene from the human, we have cut that with the help of restriction enzyme. Suppose this is the vector, we have cut it with the restriction enzyme with the same E. coli, right, E. coli 1, or here also we have cut it with the E. coli 1. Now finally we have got the sticky ends in the gene of interest, and these are also sticky end complementary to each other. We will paste it together. After pasting them, this is formed recombinant DNA, and we will put it in the host. Finally, this is done. So when we have put our recombinant DNA inside the host, it will form a lot of copies. Our gene of replication will clone, will form a lot of copies, will undergo transcription, translation to form the protein or product that we will sell in the market, right? So upstreaming process is done. Upstreaming process is done. So last 6 process of this process is bioreactor. Bioreactors are large vessels, as you can see, where we put all the raw materials that is required to that is required to form the transcription and translation to form the protein, whatever required to form the protein from the gene of interest. We will put all the raw material in the large vessels, right? We also have the temperature regulator here, right? We also have the air pumping system where we put the sterile good air, fresh air inside it. We have the pH maintainer, right? We have temperature maintainer, because we have to maintain pH, temperature, all the raw material at the optimum value, so that we will get the perfect product. Plus we are having the effluent also. We are having this particular output part, where we can just take out the product to check if our product is forming nicely, is forming correctly or not, right? So we can check it also from this outlet. So now you just see thermal jackets to maintain the temperature inside, right? So it is also used for commercial production of the vaccine, antibiotics, protein, enzymes, right? So these bioreactors. These large bioreactors are of 2 types. One is the sparged, other one is agitator. So what is sparged? Suppose this is bioreactor, it is having the fan system, right? It is having this fan system. And what it will do, suppose there's the medium. So this fan system will agitate, will actually, we also called it steerer. So steerer means we will just steer it, right? We will just rotate it so that it can give the oxygen and nutrient to all the bacteria to grow, all the organism to grow, right? And plus the foam that will form is basically, we are having the last agitator, which is not submerged in the medium. It is basically the foam breaker, right? So it is also having the foam breaker here. So these are the agitators, fan, which will mix all the things properly, right? And sparge type means it doesn't have the fan system. Rather, it is, we are just putting the oxygen in the bubble form. So why we are putting the oxygen in the bubble form? Because bubble are responsible to spread the oxygen. It will increase the surface area for exchange of gases, right? So yeah, we have these bioreactors. So sparged means we are just putting the bubble, right? We are putting the oxygen in the bubble form. Why in the bubble form? So that oxygen in bubble form, because bubble increase surface area for spread oxygen, right? Or to distribute oxygen throughout the system. So this is the sparge type. So finally, both the type of bioreactor has proper oxygen supply. One having fan, other having the oxygen in the bubble form, so that it can spread, it can increase the surface area for the spread of oxygen, right? So after we are done with the production of the product, right, finally our product has been produced using the bioreactors. The last step will be the bioprocessing, right? So sorry, what we call downstreaming process. So what is the downstreaming process? Let's see it. So the last step is the downstreaming process. So what is downstreaming process in this process? In this, we just purify the product that has been formed. So we will do the separation of the product. We will do the separation of the product. Separation of our product. Finally, we do the purification. Purify the product from the bioreactors, from the host cell. And after that, if we are just producing the drug, it will undergo so many clinical trials, right? So we will do the clinical trials. We will do the formulation. And after doing all these things, we will check the quality check for different products. So if we are selling the drug, if we are producing the drug, it will undergo more quality checks, right? Suppose we are, selling some product, forming some product that is need to be eaten. So that also require a lot of the quality check. So quality check varies from product to product, right? So all this process will done, and finally it's ready to sell it in the market. We will add the preservative and all, everything will happen in the downstreaming process, and finally we will sell it in the market, and we are done with the chapter, right? So you will be getting PDF along with these questions. So you will be solving these questions. I have put answers also here, right? So I hope you like the video. Have a great day, kids, and you will be revising everything very nicely. All the best. Have a great day.